Hello, dear colleagues, and today we continue our lectures about medical decision support systems based on artificial intelligence. And today we will talk about comparison of facts in creating digital phenotyping. And let's name what are the objectives of today's session. We will focus on the term electronic phenotyping, describe the differences between trait and phenotype. We will explain the purpose of electronic phenotyping, describe two main approaches of electronic phenotyping, the strengths and weaknesses. We'll describe imperfect labeling and how it can be used in phenotyping. In this course we already had described how to obtain data from different parts of the healthcare system, how to place events on a patient's timeline, how to obtain features and use knowledge graphs, structured and unstructured data. Now let's put this together to define this as a combination in phenotyping. First, let's define what a phenotype is. Phenotype is a set of characteristics of an organism and not only external ones. All clinically detectable characteristics such as height, body weight, eye color, hair shape, blood type, and so on are phenotype. In a clinical context, a phenotype can be defined as certain characteristics present or absent in a patient. These characteristics may also include different states. Most common example would be a disease. For example, we can say that a patient has type 2 diabetes or doesn't have type 2 diabetes. And how we can describe this as a process? It could be named as a computational procedure based on electronic medical records aimed at determining whether a patient had a condition or interest, and if the condition was observed when it began and ended. Why this process is useful for healthcare? Electronic process for creating phenotype widely used in clinical research. Electronic phenotyping applicable to observational data from research. Patient enrollment could be used in clinical trials, and calculation of quality indicators for health systems that provide public reporting also could use it. This procedure is also used in searching for similar patients and in forming definitions for large-scale interclinical research. At the first glance, everything looked quite simple. But why and when we are trying to use classification codes, for example ICD-10 or 11, that are included in patient medical record, we will face several problems. The first problem is inaccuracies that may occur when filtering or previous data or filling out a patient's electronic medical record. Erroneous Disease codes in the electronic medical record will lead to a lot of problems. For example, a provisional disease code may have been assigned by a doctor before tests or research results are available. Even if studies don't confirm the presence of the disease, the temporary code may remain on the card. The second problem is that even if all the codes in the electronic health record are correct, they may be assigned after the patient is first known to have the condition. For example, the final disease code was entered into the electronic health record only upon discharge from a hospital, and the condition was diagnosed during hospitalization. So it could be entered anytime before the discharge. And there could be different procedures for data gathering to be able to proceed with phenotyping. And we need to place all these characteristics on a chronological scale in order to determine whether the event of interest to us occurred, and if so, when it happened. We need to choose which set of features to use and which part of the timeline to consider. It's important to distinguish a trait from a phenotype. And do so, we need to understand that sign is something that has a certain objective indicators of feature. Phenotype is a fact of a presence of a combination or some interpretation of one or a set of characteristics. The difference may not be obvious, so let's look at the example. Blood pressure is a sign. It's measured in millimeters of mercury and has a specific meaning for treatment. Hypertension, it's a phenotype. Its presence can be diagnosed by a combination of blood pressure measurements in combination with other signs, such as the persistence of high blood pressure for a certain period of time, taking certain medications, prescribing certain laboratory tests, and the results of these studies. To assign a patient to any phenotype, we need to determine what features the phenotype should contain, whether these features should be present or absent. In addition, we must determine ranges of values for these features to understand whether the patient was in the condition of interest to us. The description of the phenotype must also contain certain criteria for determining the start and the end times of the conditions. Determining the start time can be a fairly simple task, but the end time of a certain condition can be a problem. For example, how to determine the end time of pneumonia. Symptoms such as coughing or difficulty breathing can continue even after the infection itself has ended. That's naming the exact time when the condition suffers from pneumonia ended is a non-trivial task. When defining a phenotype, it's very important to clearly understand the intended meanings of the phenotype definition. Will this phenotype be used to determine the patient's current condition, or it's needed to ascertain previous conditions? Both of these tasks are important, but they are not identical. It's also matters how exactly the phenotypic characteristics are recorded. Are they described in one of the spoken languages, or they are presented in the form of formalized, computer-reliable specification, or are they written using the specified language? And if you already have a definition of the phenotype, then how can you verify whether it's really possible to select the patient we need with its help? The standard of assessment usually involves comparison of screening results and medical records by experienced clinicians. This is very labor-intensive method. Moreover, it's imperfect. Please note that it's important to identify risks and outcomes of these answers for these questions. We also need to determine risk thresholds and treatment effects of these phenotypes. Thus, electronic phenotyping is a process of identifying necessary and sufficient conditions for attributes that must be present or absent and their required values in order to determine whether an exposure or outcome of interest has occurred. It's important to understand that the accuracy of phenotyping depends on the research questions and you are trying to answer them. It must be taken into account that the definitions of various conditions adopted in real practice may change or be refined over time. Therefore, electronic phenotyping is a quite difficult task, and also it's very interesting to pursue this goal. Finally, an issue to consider when assessing how well a phenotype definition based on data from one clinical site, it will be necessary to perform the same task at a secondary clinical site and to use it as a referral for phenotyping. There are two basic approaches to electronic phenotyping. Phenotyping based on rules and probabilistic phenotyping. Phenotyping as a rule-based process consists of using sets of rules to explicitly identify inclusion and exclusion criteria for the patient. Sets of rules are created by experts who reach consensus on these criteria in an internal manner. Inclusion criteria signs that what must be present in the patient's electronic medical record. And exclusion criteria is a sign that should not appear in the patient's electronic medical record. Let's use a real example to see how electronic phenotyping works based on rules. To do this, we will use the phenotype knowledge base, and it's a public repository of phenotyping definitions located at the phe.kb.org website. Let's say we decide to conduct an electronic phenotyping with developmental language disorder. We find that corresponding phenotype and analyze criteria, as we see, are description of phenotype, including specific disease codes that must be present or absent in the medical record in order for us to assign the patient to the phenotype of interest to us. That description also includes an additional age filter that screens out patients over 18 years of age. Another example of phenotype description is the type 2 diabetes phenotype. Description of this phenotype can be found at the same website. It's expressed in the form of flowchart or algorithm. The input of the algorithm is an array of records from the patient's electronic medical record. Next to the medical record is checked for established diagnosis records of taking disease-specific medications and laboratory test results. Based on the totality of these actions, a conclusion is made that the patient belonging to the phenotype suffers from a type 2 diabetes. The fact is that having only diagnostic codes is not enough to be able to use only them. It's necessary to supplement the availability of codes with prescriptions and specific laboratory values and consideration of the relative timing of events, emphasizing the need to codify prior knowledge, specifically taking into account the patient's chronological scale in this disease. It's important to remember that each data source can be useful, that combination of sources are more accurate, and that the relative value of a source depends on the disease of interest. How does one end up creating a definition of a phenotype for phenotyping based on rules? We define the data elements which should be obtained from the medical record. Use the appropriate knowledge graph to transform these data elements into specific criteria. After that, we're able to create a definition by specific criteria. Iterate against some reference standards, usually a clinical review of the full table. Let's discuss probabilistic phenotyping. It's a grouping of many multidimensional vectors available to us in the form of data, based on the use of machine learning instead of expert consensus. Of course, there will be a problem with probabilistic approach. Imagine you're the chief data scientist at the healthcare startup. You need to identify patients who have one of 50 different conditions. The work must be completed within 1 week. What could you do? The solution will be to use supervised machine learning with a training dataset with each patient explicitly labeled as having or not having the condition of interest. The training dataset is used to build a computational model that can classify whether a previously unseen patient has the disease. We calculate the number of mentions of diagnostic codes for each patient and interpret the number as the probability of the presence of the phenotype. We then use this estimated probability to group patients into those who are likely to have the condition and those who are likely to not have the condition. Often the data we can obtain from electronic health records may be incomplete or imperfect. In any case, this is not a reason to refuse to answer your research question. What can we do to still make use of imperfect data? Often the data we can obtain from electronic health records may be incomplete or imperfect, and we can begin with the selection of keywords for a contextual search for the state of interest to us. Once a set of words has been identified, we can use knowledge graphs to expand in the inclusion of related terms. We can search the samples of all patients whose records in the chronological scale contain these terms in a non-negative manner. Finally, we can train a phenotype classifier using all other features. Another method for dealing with imperfectly labeled data uses concept of an anchor. Anchor is a reliable identifier of a presence of a phenotype. The anchor is used as a labeling function to obtain large amounts of training data for the classifier. We can also track how much our classifier improves with the addition of additional anchors. Another method for dealing with perfectly labeled data uses the concept of an anchor, and it's quite efficient. Next one will be approach of Aphrodite, automatic phenotyping procedures for observation, identification, training, and evaluation. It uses standardized open source representations and vocabularies to be able to implement a tool for phenotype identification. So it's a complex tool combining different approach. And Hippocrates also considered any patient with a particular disease as a process that can be described by the rule of its dynamics. The idea of disease as a specific and complex dynamic process is the opposite of the idea of a discrete diagnosis that defines disease as a condition. Centuries of development and accumulation of medical knowledge have made it possible to determine the development trajectories of certain disease and translate them into standard medical protocols. Currently, we can conduct research on the body much more systematically than before. We have access to research at the cellular, genomic, and even molecular level. In some cases, we have access to the record of what's happening in entire healthcare systems, which means millions of electronic medical records and billions of carefully documented cases of treatment. It's accumulation of knowledge that allows us to develop a process approach to treatment, from identifying the typical course of the disease to the manifestation of patterns of disease progression depending on the set of symptoms that each specific patient will present. In other words, we can move from a general protocol for the treatment of a particular disease to an individual treatment regimen. We can adapt this regimen to characteristics of a particular patient. A snapshot of the patient's observed here and now state doesn't tell us how he ended up in this state. This condition may be a consequence of a process or processes that occurred over a long period of time. It could be preceded by other processes and diagnosis. In order for the concept of individual treatment to become acceptable and applicable, a transaction is necessary from a static snapshot of the body state to a dynamic trajectory of the disease, where many factors interact over time, including the mutual influence of several diagnoses, taking into account the phenotypic characteristics of the body and so on. The dynamics of changes in the patient's condition can be represented as a vector in a multidimensional space that combines all elements of the collected information, including very precise molecular measurements. This vector is well displayed, for example, in the form of the patient timeline, which we discussed earlier. A large number of such vectors can be grouped based on the similarity of the process. These groups phenotypes represent a certain significantly smaller than the original number of basic stereotypical scenarios. This grouping process is also called phenotyping. In addition to this technique, electronic phenotyping patient it's a process of determining whether a patient belongs to a particular phenotype that we have identified for a certain process. I hope we'll continue next with more clear definitions. Thank you for your time.